have you considered direct repeats at the end of your constructs to sort of engage with the SSA DNA repair pathway?
Yeah, so we haven't done too much work on this, although you'll notice in my constructs that there is a direct repeat on each end of the tDNA near the lock sites with a matrix attachment region. And so we're unsure at the moment whether that provides any benefit or not in terms of the excision process, it being a very similar sequence on each end. So I'm just not sure yet. It could be something worth testing. I think there's a second comment here about synthesizing up to 120 kb constructs. So if you're delivering with Agrobacterium, I do think that you will start to hit a wall past a certain point. And we've delivered successfully at pretty high rates stuff up to 60 and 70 kb, but I've never tested anything beyond that. And there are cases in the literature, for instance, in Arabidopsis floral dip, of constructs up to like 110-ish kb. So I do think, it's a long single-stranded DNA fragment, and I do think there are some physical limitations to that. So this dream of like a megabase delivery from Agrobacterium, I don't think we can achieve that. It's just not possible. I maybe I'm too much of a naysayer, but yeah,
I'll say
that's mine.
our collaborator, Roger Tillmany, who was involved in developing the gantry system. I don't want to speak too much on his behalf, but his idea is that about 80 kb, like you have diminishing returns the longer tDNA you have, but he thinks about 80 kb is kind of like the wall before it's just going to be too impractical for the amount of explains you would have to transform.